name is Dr. Laurel Kartchner, and today I'm going to talk to you about an article that was recently published from JCI titled Recipient APOL1 Risk Alleles Associate with Death-Censored Renal Allograft Survival and Rejection Episodes. So to start off with, let's provide a little background on this article. Patients that have African American ancestry often have a higher risk of non-Mendelian focal segmental glomerular sclerosis, also known as FSGS, and end-stage kidney disease. In renal transplantation, if you have mutations in the gene APOL1, especially if you have 2 copies of the APOL1 risk allele present in the donor kidney, it's known that there will be an increased risk of the graft being lost, increased risk of death-censored allograft loss. However, there's currently mixed data reporting whether or not there will be an increased risk in kidney recipients who have the APOL1 risk alleles as well. And the role of APOL1 risk alleles in non-kidney, including immune cells, has not really been examined. It's mostly been studied as a kidney-associated disease. So that's the background for this article. So for this particular study, what they decided to do was to test whether or not recipient APOL1 allelic variants would independently correlate to the transplant outcomes. And they utilized the genomics of chronic allograft rejection study data, as well as the clinical trials in organ transplantation data. So I will refer to those as GOCAR and CTOT in the course of my presentation. They looked at the genome-wide single nucleotide polymorphisms in donors and recipients from each of these kidney transplant cohorts, and then they also further characterized the population based on if they were of African descent or Hispanic descent. So this slide breaks down the demographics of the groups that were analyzed in the study. From the GOCAR cohort, there's 385 donor-recipient pairs, and from the CTOT group, there's 122 donor-recipient pairs. When you look at the genetic ancestry of the recipients, so on the top half you see the recipient characteristics. When you look at the genetic ancestry of the recipients, there's a trend toward APOL1 risk alleles being present in recipients of African American ancestry and Hispanic ancestry, which you would generally expect. And there is also a similar trend in terms of the donor characteristics. Looking further, you can see that overall there was an increased number of death-censored graft loss events in the recipients who had a higher number of APOL1 risk alleles. So with the lowest percent of death-censored graft loss being present in the groups that had 0 APOL1 risk alleles, and trending towards the highest number of death-censored graft loss events happening in the recipients that had 2 APOL1 risk alleles. And this holds true across both of the cohorts. So overall, what they showed here is that a higher number of APOL1 risk alleles in transplant recipients is associated with a higher risk of death-censored allograft loss, which is shown by the survival plots shown here. They found that the APOL1 SNP-based mismatch score had no significant effect on that death-censored allograft loss. When they adjusted for genetic ancestry, induction therapy, and donor type, there was still an increased risk in association with APOL1 risk alleles in the recipients. And when they break these down into African American and Hispanic populations, these trends still remain, indicating that APOL1 leads to a higher risk of death-censored allograft loss. So the study team created a quantitative value that characterized the recipient African American ancestry, known as rPAFR, and they found that this value was significantly associated with serum creatinine levels from 3 to 24 months after transplant, independent of any other factors. So looking at the GOCAR group, what they found was generally that a recipient with 100% African ancestry has an average of 0.75 mg per deciliter higher serum creatinine than a recipient with 0% African ancestry. They found that this elevation existed in both the GOCAR group and the CTOT group, with the values being slightly different between the two groups. They also found that an estimated GFR tended to be inversely correlated with that RPAFR, that quantitative value of African American ancestry in the recipients, in mixed models, but they didn't actually show that to be a significant difference. The team then wanted to examine whether or not APOL1 plays a role in cells other than kidney cells. And since immune cells are mediators of rejection, they decided to look at immune cells to determine whether or not immune cells APOL1 plays a role in immune cell activity. What they found was that in African American and Hispanic recipients, there was immune activation that was present in individuals with APOL1 risk alleles. They then decided to collect peripheral blood mononuclear cells from African American Hispanic recipients and determine whether or not they express APOL1, and they found that there was APOL1 expression in those participants, which is shown in panel A. They then decided to look at what immune cells might express APOL1, and so they looked in healthy individuals, so these are not kidney recipients, but instead in healthy individuals, as is shown in panel B. And what they found is that the immune cells that express APOL1 significantly are natural killer cells, activated naive CD4 T cells, and activated naive CD8 T cells. They then wanted to look at what genes were expressed within these immune cells. So they, again, looked in healthy individuals, and they found that there were differentially expressed genes involved in immune activation in individuals with the G1, G2 alleles relative to individuals with the G0, G0 genotype. And they specifically looked at CD4 and natural killer cells, that's shown in panel C. They then wanted to look at what pathways were differentially expressed in these cell types. So they looked in healthy controls again with APOL1 risk alleles, and they found that there was enrichment of genes that are associated with allograft rejection, antigen processing pathways, so HLA genes, T cell and activation and differentiation pathways, and chemokine genes. So these are all genes that are going to cause the immune system to be overly activated. And again, these are in healthy individuals, not patients. They're having differential gene expression in the cells that have that APOL1 risk allele relative to individuals who don't have the risk allele. So you see an overall immune activation in these individuals in spite of the fact that they don't have kidney disease. They also looked in both CD4 lymphocytes and cytotoxic natural killer cells and found that the differentially expressed genes included the TNF alpha signaling pathway genes and antiviral response genes as well. So next they decided to look at patients with end-stage renal disease. So this is pre-transplantation. And they utilized single-cell RNA sequencing of natural killer and activated CD4 and CD8 T cells. And they found that there was an enrichment of differentially expressed genes in these activated CD4 and CD8 T cells, as well as in the natural killer cells. They also looked prior to transplantation and found that recipients with any copy of G1 or G2 alleles showed differentially expressed genes enriched in the immune response pathways relative to patients that don't have those APOL1 risk alleles. They also found that African American and Hispanic recipients with APOL1 risk alleles had stronger responses when tested for the frequency of alloreactive interferon gamma producing PBMCs co-cultured with cell lines. So overall showing a higher level of immune activity in the presence of those APOL1 risk alleles. One major limitation of this study is that overall they did not analyze the donor APOL1 genotypes. Instead, they focused on the recipient APOL1 phenotype. Overall, this study demonstrated that the presence of APOL1 risk alleles is associated with death-censored allograft loss, and that it is also potentially associated with clinical and subclinical TCMR events up to 2 years after transplant. Even a single risk allele presence increases risk for acute and long-term graft survival, and APOL1 alleles also appear to play a role in modulation of alloimmune responses. Please feel free to reach out if you have any questions, and I hope you have a great day.